Good luck, everyone.
Thank you.
Thank you. We'll just wait a little bit for extra people to come in. It's just 3 o'clock, and then we will start with the session. Hello, and welcome to our third and last session of the virtual Discover Glow tour. We will focus today on cellular target engagement and protein degradation. We will first have a talk from Dr. Martin Gieras from UCB Pharma, followed by a Q&A session, and then we will proceed with a presentation from Dr. Christine Riching from Promega R&D, and then we will end up with the last Q&A session. First, I would like to add some practical points. Please feel free to add your questions to the Q&A box. And please also add your name, so we are really making sure that you are getting your follow-up during the Q&A session or personally afterwards. We will also make a recording of this webinar available for you, and also the slides from Christine will be made available for you afterwards. So let's now move to the first talk of today from Dr. Martin Gieras. Martin is actually working as a group leader or associate director of the drug discovery team of UCB Pharma, and they are actually supporting several preclinical drug discovery programs for small molecules and gene therapy projects. Martin, the floor is all yours. Thank you.
Okay, thank you very much for the kind introduction and for the invitation to present today. So the topic of my presentation is to show you how we are using Promega's nanobread assay technologies for target occupancy or target engagement assays. And this will be done by giving you some data on the assay development that we've done for 2 kinase projects. So the first case study that I will show you is for the NUAC1 kinase, and the second case study was assay development that we've done for the MSK1 and MSK2 kinases. So first, a short introduction to the nanobread technology itself. So as you probably know, nanobread is a bioluminescence resonance energy transfer based assay that uses the nanoluciferase as the bread energy donor and the fluorescent, in this case 680 nanometer labeled small molecule as the acceptor in the assay. How the assay is working is that you measure really the ratio of the acceptor versus the donor, and this is giving you the interaction between 2 targets of interest inside of the cells. So we mainly are using this assay technology to look into competition binding of compounds that would compete off this labeled fluorophore or labeled tracer from the cells. And this can be done in an endpoint method, or it can also be done via. And depending on the method of monitoring your tracer readout, you can have either a readout on a PIC50 of the compounds of interest or of the residence time. The advantage of this assay for us is that you can really have an understanding of how compounds are going inside of the cells and engaging the target of interest. So if we look now first into the NUAC1 nanobread assay development that we've done. First, a short introduction on NUAC1. So NUAC1 is also known as ARC5. It is an AMP-activated serine threonine kinase. It's a member of the CAMK kinase family. Its function is mainly known to induce tumor cell survival during nutrient starvation, and it is suppressing cell death. So the first thing we have done is, after with Promega, we were recommended to use their Tracer 5 for the NUAC nanobreath assays. And we took this Tracer 5 and an unlabeled analog of this, or the same tracer, but without the fluoro-4, first in our biochemical NUAC 1 assays to see how this tracer is behaving in a biochemical assay. And you nicely see here that the unlabeled tracer had a PIC50 on NUAC 1 of 9.2, whereas after labeling with the fluoro-4, we ended up with a PIC50 of 8.2. So the fluoro-4 is really affecting the inhibition mechanism of this compound, which is not an issue because you see that here you still have a very good potency with this compound. Then we were interested in the mode of action of this tracer. So is it an ATP sensitive and ATP competitive inhibitor on NUAC1 or not? And to do this, we also went first into a biochemical assay where we incubated the kinase with two different concentrations of ATP, so around Ki for the kinase, so around 50 micromolar and 500 micromolar of ATP. And you see here that there is a small shift observed with higher ATP concentrations, meaning that this tracer is an ATP sensitive kinase inhibitor of NUAC1. Then the first experiments we've done inside of the cells is really to perform a saturation binding curve of this tracer 5. And we did this in HEK cells with a transient overexpression of NUAC1 tagged with the nanoluciferase. So here you see that we went up to roughly 1 micromolar tracer, and you nicely see a saturation binding curve with this tracer 5 inside of the cells. The PEC50 we obtained here was 7.1, so tenfold less versus the PIC50 that we got from the biochemical assays. You see the millibreath ratio is very, very high, so very good assay with a very good assay window to work with. As a next step, we wanted to know what the kinetics is inside of the cells and where we would have a steady state measurement in the assay. And therefore we did a kinetic live cell measurement where we added the tracer to the overexpressing cells, and we really monitored over time what's happening with the signal. And you see here that after around 40 minutes of incubation with the tracer, we really nicely obtained a steady state. And this both at the EC50 of the tracer and the EC65 of the tracer. So this is raw data, this is the background, and this is the background corrected data, where you nicely see that with the EC65 we have a slightly higher breath ratio than with the EC50 as expected. So this was all very nice to see. Then we were a bit wondering why we had this shift in between the PIC50 of the biochemical assay and the cellular assay. And one of the reasons that we thought about, because we knew the tracer was an ATP sensitive tracer, we know that the ATP concentration inside of the cells is different to what we use in a biochemical context. So to understand this further, we've done two experiments. The first experiment where we compared the behavior of the tracer in a saturation binding experiment between permeabilized and non-permeabilized cells. So the data you saw before was on the non-permeabilized cells where we had the PEC50 of around 7. So that's what you see here again in the blue curve. If we permeabilize the cells, so really break the cell membrane with digitonin or freeze-thaw, you see that the PEC50, the saturation binding, is really increased, and we also have almost a tenfold shift again into our PEC50. So here we come more close to what we had from the biochemical assay. So then we thought, okay, let's go one step further and really confirm then that in the permeabilized system, what we expected is once you permeabilize your cell, you also would dilute the ATP that is inside of the cells because of the permeabilization and the distribution in the buffer that you use for the permeabilization. So therefore we decided to add again ATP inside of the mixture and see if we would have again the shift observed in our PEC50s for this tracer 5. And that's what you can see here in this curve. So this is non-permeabilized cells versus permeabilized cells. And you nicely see, depending on the ATP concentration, we are shifting again the PEC50 that we observed. So this was all very nice and in line with what we were expecting, how the assay and also how this tracer is behaving from the biochemical to the. Then we've done a few experiments where we wanted to see if this assay is also suitable to do real kinetic measurements, eventually to understand the residence time of some of our compounds in the cells on NUAC. And to do that, we applied the Motulsky and Mayen model for one-state competitive binding evaluation. So here you see, not to bore you too much with all the calculations behind, but here you see the association constant that we calculated for the tracer 5 and the dissociation constant that we calculated for the tracer 5, and an example of a UCB compound that we used in this competition binding assay to measure really the KD of this compound inside of the cells and also the half-life that the compound has on the NUAC1 kinase. So this was all very good to see and useful as well for us to understand further the mode of action of the compounds that we are interested in. During the time we were doing the experiments, we had access to several NUAC1 kinase inhibitors, and we evaluated a subset of these NUAC1 kinase inhibitors in a biochemical assay and compared the PIC50s we obtained in the biochemical assay with what we obtained in the nanobread assay. And here you see the correlation between the data that we received. So this is the enzyme assay PIC50, this is the nanobread assay PIC50, and you see very nice correlation observed between biochemical and cell-based nanobread data that we obtained. So this was all good news for us to work with this assay. Then the second case study is very similar to the first case study, but I still want to show it because we've done we've had some. So MSK1 and MSK2 were two other kinases that we are interested in. And to give you a bit of background on these kinases, so MSK is a mitogen and stress activated kinase. It's a nuclear serine threonine kinase. It is a special kinase in a way because it has two different kinase domains, an N-terminal kinase domain and an C-terminal kinase domain that are fused together. The N-terminal kinase domain is part of the AGC kinase family, and the C-terminal kinase domain is part of the CAM kinase family. It is responsible for cellular stress reactions, and it has an anti-inflammatory role upon activation. Here, Promega also recommended to go with this tracer 5 for the nanobread assays, and we did exactly as for the NUAC1 assay development. Is first we checked the PIC50 of this tracer 5 and the unlabeled analog of that one in a biochemical assay on MSK1 and MSK2, and here you see the data for MSK2. So for MSK2, the unlabeled tracer had a PIC50 of 7.9. It is ATP sensitive, same as here for the labeled tracer. The PIC50 is half a log lower, so not a tenfold shift here, but a half a log difference between labeled and unlabeled, and also ATP sensitive in this case. So here the shift is much clearer because of the behavior of the enzyme. But that was in line what we expected. Then same as for NUAC, we did the transient overexpression of MSK1 and MSK2 in HEK cells. and did some saturation binding experiments with this TRACER5. And here you see the data of the TRACER5 with 3 different cell densities in the MSK2 assay. So the PEC50 is not much affected by the cell density, so it's very slightly affected, which is also expected because it's the ratio that you are measuring with this assay. And you see here the window is slightly less than what we obtained for the MSK for the NUAC assay, but still useful to work with. So in this figure, you really see the comparison of the saturation binding curves of the 3 different kinases that we use this TRACER5 on. So here, this is the NUAC1 that you've seen before with the assay window of almost 18-fold. For MSK2, our assay window was around 6-fold. For MSK1, you see here the effect was, or the TRACER5 was, to our opinion, not really useful. It's really shifting even more the saturation binding kinetics, and we came very close, as you see here, to a negative control that we use, so just a nanoluc on its own overexpressed in the cell. So for us, this TRACER5 was not useful for the MSK1 kinase, but we used it for NUAC and MSK2. Again, steady state binding of this TRACER5 on MSK2. So in this case, almost after 30 minutes, we could nicely see a steady state binding. So we decided for both NUAC and MSK2 to run the assays more or less after 2 hours of incubation with the tracer, so that we were nicely in a steady state mode of the assay. Again, same correlation between enzyme and nanobread for MSK2 with the components we had at hand, and in general there was also good correlation between the PIC50s that we obtained in both assay formats. Then we were really keen on also having a tracer for MSK1. So what we decided to do is to try to make our own tracer. And there we had some contact with the chemist in the Promega team to help guide us on how to do this labeling of a compound that we were interested in, to see if we could kind of change it to really a tracer that we could use for nanobread assays. And here you see this is the unlabeled compound. So it was really a very potent compound that we selected. So with the PIC50 around 8.4 on MSK1. And then we labeled it with the same fluorophore than it's used than I showed before for the NUAC experiment. And you see here that after labeling 2 different compounds in this case, or 2 different positions, that's what we've done. You see that the compound is still, after labeling, active in the biochemical assay on MSK1, but again is dropping potency slightly. So for this labeling scheme, we were dropping almost 1.5 log. For this labeling, we had but still, for us, with a PIC50 of 7.5, good enough to start some assay development to see if we could also use this novel tracer or potential novel tracer into the nanobread assay. And there it was a bit disappointing because we were never really able to see to get this assay running with this novel tracer. So in this case, we incubated both the nanoluc control cells that I showed you before, as well as MSK1, MSK2, and the NUAC transient overexpressing cells. And you see that there is really no big difference here in the nanobread signal that we obtained. And what was very curious is even that on the nanoluc negative control that we used, we really had a nanobread millibread ratio of around 20. So and again for both of the tracers, so for tracer 1 and for tracer 2, it was even much higher. Then anyways, we tried to do a competition binding experiment with known NUAC and MSK inhibitors, and you see that we were not able to remove or compete this tracer of either NUAC or MSK1 and MSK2. So for us, we stopped trying to develop our own tracer for these assays. So we gave it a shot, but we did not move forward with this one anymore. So then, in general, I think we can say, or from our own experience, this nanobread assay technology is very feasible for compound testing in a screening context, as you've shown that we have been doing, but also for more detailed mode of action studies as kinetic binding and measurement of residence time. Assay window obviously varies between different targets. So we had 2 good targets, 1 more difficult target in this case. We've shown that kinase tracer 5 is ATP sensitive in NUAC1 and MSK2 biochemical assays, and the potency shift that we observed for ATP sensitive kinase inhibitors between the biochemical assay and the cellular assay is, from our analysis, really probably highly likely due to the higher concentrations of ATP that we have in the cells versus how we run our biochemical assays. And then in general, we observed a very good correlation between the biochemical PIC50 values and the cellular nanobread PIC50 values for the ATP sensitive kinase inhibitors we were working on. And with this, I want to conclude my presentation, and I'm happy to take your questions.
Thank you, Martine, for this really nice presentation overview of your data on NUAC and MSK1 and MSK2. I'm just waiting if there are some questions coming in. It seems that your presentation was so clear. There were no questions yet.
Okay.
Is there any other project, future project, that is in the pipeline that you're thinking of with nanobread? I mean, you can't disclose your targets, of course, but is this something you will keep on using in your...
I mean, seeing, yes, the technology in itself, if we have interest to kind of do this cellular target engagement method, if there's a good tracer available, then it's really an assay we keep in our minds, in our head to use to support the projects. Yeah.
Okay, so I think then maybe we can move already to the presentation of Christine. Yeah, most questions are coming in while we are jumping to the other presentation, but then we can answer after both presentations. Oh, I see maybe just one question coming in. Looking to it. Would it be an issue to have no correlation between biochemical and in-cell PIC50? And what about cell permeability?
Yeah, so that's a good point. So you can have a discrepancy between your 2 assays, and cell permeability can be a reason why you have discrepancy. But by doing the assay with permeabilized and non-permeabilized cells next to each other, you can try to get an answer to that question. But it's not really mandatory to have a correlation. It's, for us, it was interesting to see that there was a good correlation for most of the compounds. Yeah. Because that's also indicative of the permeability of the compounds.
Yeah, thank you. Christine, is it possible for you to maybe share your screen? And in the meantime, I will introduce you. So thank you, Martine, for giving your presentation. When other questions are coming in, we'll come back to after Christine's presentation. So we'll now move to the second talk, and this will be given by Dr. Christine Riching. She's actually working as a senior scientist at Promega R&D, and they are developing approaches to monitor functional mechanisms of proteasome-mediated protein degradation. So thank you. Christine, the floor is all yours.
All right, thank you for that introduction, and thank you, Martine, for that excellent talk. So I'm going to be talking today about how we can characterize the efficacy and mechanism of action of degradation compounds. And specifically, I'm going to be talking about 2 case studies, as I'll get to in a moment, but what we wanted to do is try to assess both the degradability and the selectivity of key therapeutic targets. So first, to talk about targeted protein degradation a little bit in general, this is a relatively new and exciting therapeutic modality in drug discovery. This utilizes heterobifunctional compounds. They're often called PROTACs or proteolysis targeting chimeras, but they are basically a dual warhead small molecule, and they consist of the binding ligand to both the target protein as well as a ligand which binds and recruits an E3 ligase. So these compounds, these PROTACs, must first cross the cell membrane and then engage both the target protein and an E3 ligase to form what is called the ternary complex. And this ternary complex must exist in the context of a much larger complex that is an active E2, E3 ligase complex, which will then transfer a ubiquitin molecule to the target protein of interest. And upon successive ubiquitination of the target protein, this will be recognized by the proteasome for degradation. So why is there interest to develop degraders as opposed to inhibitors? And the primary reason that we've seen with many successful examples thus far is that they can exhibit much greater potency than classical inhibitors. And this is primarily due to their catalytic mode of action, where we can improve upon efficacy while using a much lower dose of the drug. There's also reduced likelihood for resistance to develop. And because the protein needs to be first resynthesized by the cell, there's the potential for extended durability of this class of molecules over inhibitors. There's also the potential to now disrupt larger complexes or scaffold proteins if we just remove one of the protein components. And also the main promise here is now that we only really need a binder to a target protein anywhere on the surface of that protein and not necessarily with high affinity in order to potentially degrade that target protein. So this really expands the possibilities for degradation, and now perhaps the excitement that is in this field is basically around the potential to now drug the undruggables. So I'm going to talk about 2 case studies today. And the first is really looking at these proteins that we might consider as undruggable targets. How might we actually study them for degradation in the absence of a target-specific PROTAC or any target-specific binder? And so for this, I'm going to talk about using halotag fusions coupled with haloprotac3 for degradation, as well as looking at protein degradation phenotype that results. And in the second case study, this is a story about profiling degradation selectivity across a broad family of target proteins. So for this, I'm going to talk about profiling degradation selectivity across the CDK family with a pan-kinase PROTAC, and talk about some mechanistic insights that we uncovered regarding the differential regulation of degradation across this family. So to start out thinking about these undruggable proteins, they are undruggable because there is not necessarily a specific binder from which we can build a PROTAC compound. So we want to be able to study these targets for degradation, but this presents a challenge. It's often known what the knockout or mutant phenotype is, but we have to be careful with some of these experiments. They can be challenging to parse out target-specific effects versus off-target global effects. And what we really want is a way to selectively remove at the protein level a target from the cell, and potentially in a conditional fashion, such that we can remove or bring back those protein levels and assess the phenotype of that loss, as well as whether we can induce a rescue. But then the other challenge here is that not all proteins will be accessible for PROTAC-mediated degradation. So before we embark on a campaign to design PROTAC molecules for any given target, we might first want to assess whether it's even possible to degrade a given protein within its cellular compartment. Is it accessible for degradation by an E3 ligase, and so on? And so these are questions that we have sought to try to address using halotag fusions with haloprotac3. So haloprotac3 is a compound that was developed by Craig Crews. It consists of a chloroalkane that covalently binds to a halotag. And this chloroalkane is coupled to a ligand which will bind to VHL. So this will recruit a VHL E3 ligase to halotag it will result in ubiquitination of halotag and degradation of halotag, as well as whatever protein halotag is fused to. So halotag is a 34 kilodalton protein fusion tag. It can be appended to the N or C terminus of any given protein. And to study the phenotype with this approach, we use CRISPR-Cas9 to tag endogenous proteins with halotag. And to illustrate that approach here, we typically use a plasmid donor template that contains the halotag sequence. And then we have 500 bases of homology to our target gene of interest. And then we use Cas9 coupled with a guide RNA complex, and we perform the editing in cells. And typically, because of the size of this tag, this occurs with relatively low frequency, but we can leverage the fact that we have a number of different fluorescent halotag ligands already developed, such as this one, the Janelia fluor 646 halotag ligand. So this will, again, covalently bind halotag in the cell. And we can see this fluorescent signal indicative of our knock-in. And so then what we can do is perform FACS to sort for halotag positive cells. As you can see here from this histogram, we don't have a very large population of positive cells, but we can still very readily sort those and generate clonal cell lines from this. And what we've done here is we've confirmed our clonality. We've also confirmed proper localization. I'm showing as an example here a knock-in to BRD4 in HEC293 cells. And again, we've labeled the cells with this Janelia fluor ligand, which is fluorescent, and we can image via confocal microscopy. But as you'll also notice, we knocked in a tandem tag here to BRD4. So we knocked in HYBIT as well as halotag to BRD4. And I'll talk about this more in a moment, but this is a way to now look at luminescence. And so using luminescence microscopy, we can also confirm and validate our clonal population here, as well as the localization to the nucleus. I want to take a quick step back and talk a little bit about this HYBIT peptide. So this is a binary complementation strategy. And so basically this peptide is a small 11 amino acid peptide, which has high affinity for large bit, which is a 17 kilodalton protein. And due to this high affinity, these fragments will spontaneously complement in cells and will reconstitute a functional luciferase protein. And so what we essentially have in the cell is our target protein fused to a functional luciferase reporter, and we can use this as a readout of protein levels. So if we treat the cells with a PROTAC, we can follow the degradation of the protein simply by following the loss in luminescence. So the workflow is very straightforward here. We have our edited cells from the previous knock-in step we performed, and we just plate them. We add a substrate, and then we can read luminescence on a plate reader. And we can do this as either an endpoint fashion, or we can monitor luminescence continuously over the course of 24 hours. And I just want to highlight the reader that we use most often for these experiments. This is the GLOMAX Discover. It's a very sensitive instrument for detection of endogenous protein levels with a very large dynamic range. But essentially what we get from these experiments is a complete cellular degradation profile, where we have an initial degradation phase and we see loss of the protein levels over time. And if we follow this kinetically over time, we can see how long the protein levels remain low and at what point they start to recover back to their baseline. And there are a number of quantitative parameters that we can get from these profiles. So if we look at the initial degradation portion of the curve, we can fit that to a single exponential decay and quantitate a rate of degradation. We can also calculate this plateau, which we also call the degradation maximum or Dmax, and we can also look at how long this degradation persists or the duration of degradation. So using this tandem tag approach, we can use halotag to induce degradation of any given target protein, and then we can use HYBIT for detection, luminescence detection of that degradation. So to illustrate that here, we have done this now for a variety of different targets in various cellular compartments. So this is initially just looking at a cytoplasmic localized protein where we've knocked in the tandem tag, halotag, and HYBIT. And then we treated with a titration of HaloProTac3 and did our extended time course kinetic degradation studies. And you can see over 24 hours, we see very rapid and sustained loss of this cytoplasmic protein. We also looked at other cellular compartments, nuclear localized protein, as well as a mitochondrial membrane protein. This is a single pass transmembrane mitochondrial membrane protein. And we see very similar rapid and potent degradation kinetics across all of these different proteins. But we did quantitate the rate of degradation as shown here, as well as the degradation maximum to look at potency. We see very similar rates and potencies for all of these proteins. So this really indicates that the halotag itself is likely the substrate for degradation. The halotag is likely what is getting ubiquitinated, and the target protein is simply along for the ride. So we can very clearly show that this approach is amenable for many different targets in different cellular compartments. But we're also looking to expand upon this as well. But now I want to talk about how we might assess phenotype as a consequence of the degradation. So for this, we turn to a different system in beta-catenin in particular. And we chose this model system because there's no known PROTAC for beta-catenin, as well as very limited binders for beta-catenin. And so we wanted to see how well this could work using this approach. So beta-catenin is a primary component of Wnt signaling, but it also has many other functions as well. It exists at the cell membrane as it's involved in cell-cell contacts, but it also exists in the cytoplasm where it is in direct response to Wnt signaling. So in the absence of Wnt signaling, beta-catenin is phosphorylated and degraded. However, when Wnt signaling is active, beta-catenin levels are stabilized and it translocates to the nucleus where it interacts with TCF-left target genes. So we generated a knock-in of the tandem tag halotag HiBit to the C-terminus of beta-catenin. And we initially just validated our clonal population of cells by observing the response to Wnt stimulation. So we did a titration of Wnt3a ligand, and we could see nice stabilization of beta-catenin protein levels. We could then also observe very rapid degradation at all levels of Wnt stimulation when we treat with haloprotac3. So at all levels of Wnt stimulation, we see rapid degradation of beta-catenin. We also confirmed by luminescent imaging the proper localization as well as the degradation of beta-catenin. So here we see localization is primarily at the membrane, as we would expect, but also there's some intracellular cytosolic pool as well. But if we treat with haloprotac3, this is showing a 2-hour treatment with haloprotac3, we start to see that dramatic loss in the signal, which is indicative of the protein being degraded. So to look at the phenotype, we were interested in looking at the consequence of activation of TCF target genes. So to look at this, we introduced into these cells via transient transfection a TCF firefly reporter construct. And in the presence of Wnt3a ligand, we can see nice activation of this reporter, although in the presence of haloprotac3, we see very little activation of this reporter. And this directly correlates with the reduced levels of beta-catenin because we've degraded it with haloprotac. So we think this is a really nice example illustrating how we can use this technology. We can use halotag and haloprotac3 to study not only the degradation of proteins that might be difficult to target because there aren't any known binders for these targets, but we can also use this as a way to understand the resulting phenotype. So now I'm going to transition into the second case study. And as I mentioned, this is a story that we looked at where we wanted to try to understand degradation selectivity across a panel of proteins. And so for this, we turn to the CDK family of proteins using a pan-kinase PROTAC. So the CDK family consists of 21 different members, and they all have various roles, various functions in the cell, but they're kind of categorized by different functional subclasses within the family. So the most classically known are the cell cycle associated CDKs. These are CDK1, 2, 4, and 6. And then there's also the CDKs that are very heavily involved in transcription. And these are CDK7, 8, 9, 12, and 13. But then there are another group of CDKs, oops, So the remaining CDKs are relatively understudied compared to the cell cycle and the transcriptional associated CDKs. There's not as much known about these, and they don't necessarily fit nicely into these other functional subclasses. So I've just denoted them here as understudied. But we generated HiBiT CRISPR knock-ins to 16 of the CDK family members in HEC293 cells, and I've denoted the terminus that we tagged each protein family member here, as well as the zygosity that we established by Sanger sequencing. But what we wanted to do was profile degradation across this family with this PROTAC shown here. This is TL12186, and this was developed by Nathaniel Gray's group. So this was shown in mass spec studies to be capable of degrading, I believe, 26 different kinase targets, 8 of which were members of the CDK family. And this was shown in MOLM and MOLT4 cell lines. So we generated this panel of HiBiT CRISPR knock-ins, as I mentioned, and initially we just wanted to look at the relative expression across this family of proteins. So just by looking at the relative light units, or the RLU, we can see almost a 2 order of magnitude range in expression across this family. And we see, in general, the most abundantly expressed proteins were the cell cycle and some transcriptional associated CDKs. But now looking at degradation, so we initially just wanted to hit the cells with a single concentration, a 1 micromolar concentration of this compound. But then we wanted to look at an early, so just a 6-hour time point to see how much loss we could observe across the family. And you can see, basically, we do see some amount of loss with each family member, where in some cases we see pretty dramatic loss at 6 hours, but in other cases we're only starting to see some loss. If we look further out to 24 hours, now we see a bit more dramatic loss across the entire panel, but still we see a pretty wide range in responses. There's definitely not a consistent amount of loss across this family. And we also observed that there's no obvious correlation with the extent of degradation to the expression level of each individual CDK. But we did go ahead and we performed the kinetic profiling experiments. So this is looking at degradation over the course of 24 hours with the titration of this compound. And we did this for all 16 CDK targets we studied. So I'm not going to go into detail talking about all of these different curves, but I just want to give you a sense of the broad range of responses that we observed, where some CDKs were very potently degraded with this compound, as in the case of CDK12 and 13, where you can see even at the highest concentration, we start to see what is known as this hook effect, where we start out competing the ternary complex formation by favoring the binary complexes, and that is indicated by a slowing of the degradation rate at high concentrations. But we also see, in some cases, very weak degradation. So I'm just going to point out CDK6 as an example. So you can see even at a 1 micromolar concentration, we're barely degrading 25% of that protein. But how do we actually kind of tease out information from all of these responses? So to look at this more closely, I've plotted here from those profiles that I showed you in the previous slide, just the 1 micromolar trace, and then I've grouped them by the functional subclass. So we have the cell cycle associated CDKs, the transcriptional, and the understudied. And you can clearly see by looking at the profiles in this fashion that the cell cycle associated CDKs clearly show much weaker degradation, more incomplete degradation, while the transcriptional and understudied CDKs show more of this classic degradation to a plateau sort of profile that we are accustomed to seeing with PROTAC compounds in general. So we see much more rapid degradation and more sustained and complete loss of the CDK targets. So we also looked at the degradation maximum. So from the full concentration curves in the previous slide, we calculated the degradation maximum across each concentration, and now we could calculate potency for the transcriptional and understudied CDKs. And we could see for several of these, we could get down into the single-digit nanomolar. But the cell cycle associated CDKs, again, these showed much weaker and incomplete degradation. And we wanted to understand why this is. Why does this subclass of CDKs seem to show weaker degradation with the compound? So In order to address this, we decided to focus for the remainder of these experiments on CDK2 as its particular relevance as an important therapeutic target and as a member of the cell cycle associated CDKs. So CDK is, of course, a highly regulated cell cycle associated kinase. It has many different active and inactive forms, as well as different phosphorylation states, and it's involved in multiple different complexes, as well as protein partners, cyclin protein partners. And so the primary cyclin partner in G1 to S phase is cyclin E, and the primary cyclin partner in S to G2 phase is cyclin A. So we wanted to understand whether there's potential for differential degradation with respect to the phase of the cell cycle. And if so, is there perhaps a mechanistic difference that we can understand with respect to degradation within the different phases? But first, we wanted to show that this loss that we're observing of CDK2 is dependent on a PROTAC mechanism and is also dependent on proteasome-mediated degradation. So to look at that, I'm showing the profile, the degradation profile in the presence of the TL12-186 PROTAC only. But then we also performed that same titration in the presence of a molar excess of lenalidomide. So we're essentially inhibiting the formation of ternary complex, and we see basically we've halted degradations. We no longer see the same extent of degradation at the highest concentrations. And we can also basically inhibit the degradation when we treat with MG132. So this shows that indeed this is occurring by a PROTAC-driven and proteasome-mediated mechanism. If we look at treatment with bacillomycin, so this is an autophagy inhibitor, so we can still see when we treat with the titration of the PROTAC, we can still see robust or modest, I should say, degradation of CDK2. So this confirms a PROTAC-mediated mechanism. It's dependent on the proteasome. We don't see any difference if we inhibit autophagy. If we look at just the parental kinase inhibitor alone, so the inhibitor that this PROTAC is based off of, we do see some modest loss of CDK2 levels over time, but this might be due to some transcriptional feedback just as a consequence of the inhibition. So the next question we wanted to ask is whether TL12-186 mediated degradation is regulated or influenced by the cell cycle. So to look at this more closely, we attempted to arrest the cells in different phases of the cell cycle with various treatments. So we have untreated cells, we have serum-starved cells, we have a compound that we treated the cells with to induce S-phase arrest, and then we also treated with nicotazole to arrest cells in G2M. And after performing these treatments, we then added a titration of the PROTAC compound to observe degradation. And interestingly, we saw the most robust degradation of CDK2 in serum-starved cells compared to the other treatments, where looking at S-phase arrest or arrest in G2M, we see very little to no degradation of CDK2, which looks like some of the other control treatments with lenalidomide or MG132, or even the parental kinase inhibitor that we showed before. But also, if we just look at the expression level after treating with these various cell synchronization treatments, we don't see any obvious correlation of the degradation to the expression level. So now I want to take a step back and talk about how we might try to understand the mechanism. So how we can investigate the basis of this difference that we see with respect to the cell cycle. So for this, I'm going to talk about nanobreadths, and we've already had a nice introduction to this technology from Martine. So basically now we can use our HiBit CRISPR endogenous protein as our luminescent donor for energy transfer. And to look at a protein-protein interaction, we transiently introduce into the cell a halotag fusion. So this would be a halotag fusion to, in this case, cereblon to look at ternary complex formation, and this would serve as our fluorescent acceptor. And then upon interaction of these two proteins, we would see a gain of signal indicative from the energy transfer. But similarly, we can also look at protein small molecule interactions. If we have a fluorescent tracer molecule which binds to our target protein of interest, we could then displace this tracer from the target with our compound, our unmodified compound. So we wanted to ask whether the PROTAC bound to CDK2 with similar affinity in all phases of the cell cycle. And so to do that, we looked at target engagement, where we had a tracer that bound to CDK2, and we could displace that tracer with this PROTAC TL12186. And if we performed the same treatments to induce the cell cycle arrest, we see that we could displace the tracer with very similar binding IC50s, or displacement IC50s as shown here. So this indicates that CDK2 binds the PROTAC with very similar affinity in all phases of the cell cycle. But what about ternary complex formation? So to do this, we now transiently introduced into the cell a halotag fusion to cereblon, and we can look at the induced ternary complex formation after doing these same treatments to arrest the cells in different phases of the cell cycle. So if we treat with 1 micromolar TL12186, we actually found that only in untreated or serum-starved cells do we see appreciable ternary complex forming. Whereas in nicotazole or S-phase arrested cells with this compound, we see very little to no ternary complex formation. So just to summarize here, what we've shown with this study, we've seen using this panel of targets across the CDK family and profiling degradation with this PROTAC, we see a broad range in responses to this degrader. And we see actually that the cell cycle associated CDKs show this very interesting limited degradation. And we could also identify when we more closely examine CDK2 that CDK2 is differentially degraded with respect to the cell cycle and occurs primarily in G1. We also showed that these differences are not due to differences in target engagement. So there's no difference in the ability of this PROTAC to bind CDK2 in the different phases of the cell cycle, but rather the impact is on the ability of the ternary complex to form in different phases of the cell cycle. So this really suggests that potentially the states, the activation states, the post-translational modifications of CDK2, or perhaps the different protein partners that CDK2 is bound to in the different phases of a cell cycle might regulate its accessibility in the context of ternary complex formation with cereblon. But another question that we could ask is, are there cell cycle dependent differences then in the ubiquitination? We might expect this to be the case. We are in the process of looking at this, but I just want to show you that we can observe in untreated cells where we know that ternary complex can form, we do see CDK2 ubiquitination upon treatment with this PROTAC. So this correlates very nicely with the data that we showed with respect to CDK2 degradation. But the take-home message here that I want to point out is that there is perhaps now this potential to engineer degradation of a specific target pool or a subpopulation of a target through the choice of an E3 ligase. So if we think about proteins that are localized to different compartments of the cell, or perhaps are engaged in different binding complexes at any given time, does this now present a mechanism for which we can engineer selectivity of different subpopulations of targets? So I just kind of want to introduce that as food for thought, and then acknowledge all of the people who were responsible for this work and who contributed to this work at Promega, as well as our collaborators and Craig Cruz in particular for development of HaloPROTAC3. And with that, I'd just like to thank you all for your attention and would like to open it up now for any questions.
Thank you, Christine. Thank you for this very nice presentation. Let's just move to some questions we received, and maybe I would like to go back to the first presentation from Martine first. So Martine, there was a question from an attendee. First of all, they want to thank you very much for your excellent presentation, and they wanted to ask what you have in mind as being the biggest advantage of using the nanobread assay over a biochemical assay.
Yeah, I expected this question, honestly. So, yeah, so I think the biggest advantage is that you really can understand how a compound is working more into a cellular context. So, given the fact if it's going over the cell membrane, and in our case, we had a good correlation between biochemical and cellular target engagement in a way, which is for us kind of for a screening context. We mainly rely our SAR activities on the biochemical assay. But if you would have for many understood reasons, kind of a shift between what you see in a biochemical and into a cell assay. This cell assay is an important assay as well to drive your project forward and to see what target engagement is in a cell context. So it's a bit dependent, I would say. Yeah.
Okay, thank you. Now a question maybe for Christine. Is there a pool of beta-catenin that is resistant to degradation? The attendee said there still seems to be some signal in your image and also still some reporter activation.
Yeah, that's a great question. So that's actually a question we're very interested in looking at further. We just have not yet had a chance. So we would like to look at these experiments using imaging. And our initial data, we do actually think that there is some population of beta-catenin that is not being as efficiently degraded as some other populations. So we're very interested to see if we can parse that out. We think it actually might be a little bit more of the membrane fraction that is perhaps more resistant to degradation, but at this point we don't know. But yeah, and then in response to the reporter activation, yeah, we definitely do still see some reporter activation, and that perhaps we could eliminate if we used a higher concentration of the PROTAC, which we did not do in that experiment. But perhaps we're just not completely degrading everything at that concentration. So there are some potential additional experiments we could do to look at that further.
Mm-hmm. Thank you. Another question, Christine. Do you have any phenotypical results of degradation of beta-catenin? And yeah, another question is coming with, is beta-catenin degradation a viable strategy in cancer therapy?
Yeah, that's a great question. I guess I don't have a sense of whether it would be a viable strategy for cancer therapy, but we used it as a way to illustrate how one could use this approach, because it is a target that is a notoriously difficult target to drug. So that was the main reason we chose it, not necessarily because it would necessarily be a viable strategy in that context from a therapeutic point of view. Does that answer the question, or was there another part to that question?
Yeah, there was also another part, and I would like to add, is synthesizing a PROTAC that is able to bind directly to beta-catenin one of your prospects?
Could you perhaps repeat that question one more time?
So synthesizing a PROTAC that is able to bind directly to beta-catenin one of your prospects.
No, so we are not, we don't typically make PROTACs. We are developing tools to characterize them and understand their mechanism of action in cells. So we're kind of using our luminescence-based technologies to assess degradation, but we are not developing PROTACs ourselves.
Yeah, I think that answers his or her question. Yeah, we're coming towards the end of this presentation, so I really want to thank you, Christine, and also really Martine from UCB Pharma a lot for your nice presentations. I'm certain, yeah, there were certainly extra questions that will come in, and then we will follow up with you separately afterwards. I just want to thank everyone for your attention, and if you have any questions that you would like to reach out to us directly, please don't hesitate to reach out to us directly. You see the contact details on the screen. So yeah, we will also make the recording of this webinar available and also the slides from Christine. And thank you again for joining, and I wish you all a nice day and stay all safe and well. So thank you. Bye.